Let's take a moment to think about what chromosomes are and organize the way that we think about them in biology courses. Chromosomes are thread-like structures of double-stranded DNA and protein. You see that I'm highlighting protein in red there. That's a often forgotten component of chromosomes that work together to store genetic information. Double-stranded DNA can be drawn kind of like this, with two individual strands of that DNA sort of winding around each other in a long double set of lines. Another way to think about that is two parallel lines that run in opposite directions. Okay, we use a 5 to 3' notation to indicate direction on DNA. And in between those lines, you get your bases, which are complementary to one another. So if I have ATCG running in this direction, the complementary strand will have CGAT running in the opposite direction to complement that. Okay, these are two different ways to think about the structure of DNA. Remember that I said DNA on chromosomes actually includes proteins. And eukaryotic cells, those are proteins like histones, which I'm just showing as a little circle with an H in it here. And the reason organisms have evolved this relationship between proteins and DNA is so that the DNA structure itself can be dynamic. It can change shape and it can move around the cell. The overall structure of a chromosome depends on which cell type we're talking about. In biology, there's two main divisions of life: prokaryotes, which are things like bacteria, which typically have small circular chromosomes. So I'm just drawing the two strands of the DNA there. And these are typically smaller cells, again, things like bacteria. And that chromosome occurs in the cytoplasm. Okay, so prokaryotes, of course, do not have membrane-bound organelles. Contrast that with eukaryotes, which have larger cells, typically. These can also be multicellular organisms like plants and animals and fungi. And these have a nucleus, which houses almost always linear chromosomes. Okay, here I'm drawing the condensed form of that linear chromosome, but in reality, this is just a single molecule of DNA with a lot of proteins on top of it, and we could actually unpack that DNA, that double-stranded DNA, into one long strand. Actually, it's a double strand. And there are other organelles in eukaryotes, of course. Oftentimes, you'll find things like a mitochondria. So down here, we had our nucleus. Here is our mitochondria. And some of these organelles, like mitochondria, are thought to be the evolutionary legacy of an engulfed bacterium billions of years ago. And because of that, the DNA that still exists inside that mitochondria is itself circular. The chromosome is circular inside there. Okay, and these are much smaller chromosomes than the linear ones you would typically find in the nucleus. Another major difference between prokaryotes and eukaryotes is how these cell types divide and the differences therein, paying particular attention to chromosome structure. In prokaryotes, which have one or a few circular chromosomes, before cell division can occur, the DNA in that chromosome must duplicate through a process called DNA replication. We started with one copy of that chromosome, and we end up with two copies of that chromosome. This will then allow for binary fission, in which case that single cell that contained that chromosome before has the ability to split off into two distinct cells, both of which have the genetic information of the parent cell that gave rise to those. Okay, so we call that process binary fission. And that is the simplest type of cell division. With eukaryotes, there are two main types of cell division, and you've all heard of mitosis and meiosis. Mitosis is the process by which one eukaryotic cell divides to create two identical daughter cells. This is useful for things like organismal growth. So as a zygote proceeds to adulthood in a multicellular organism, this is how that organism gets bigger and grows new tissues. It's also something that happens through the process of repair. So as you get a cut on your skin and that tissue must grow back, mitosis is how those cells are filling back in. It's also useful for the process of renewal. So as the linings of your digestive system dies off and those cells must be replaced from stem cells, all of that's happening through mitosis. Contrast that with meiosis, and meiosis has a much more specific role in cell division. Instead of one cell dividing into two identical daughter cells, we have one cell dividing twice, 2 rounds of cell division, which leads to 4 non-identical gametes. And this is not for growth and not for repair. This is specifically to create the cells that will be useful for reproduction. Going back to mitosis, you don't have to be a math major to realize that something looks a little odd here. How do you take a cell that has 2 copies of its genome? That's what this 2N means. So most eukaryotic cells are diploid, which means they have 2 complete copies of the genome. How does a cell like that divide, creating 2 different cells, and at the same time maintain the exact identity that it had before? How do you take 2 of something, divide it, and end up with 2 sets of 2? Well, the answer is there's a hidden step that I didn't draw, and that sometimes textbooks fail to draw depending on what particular topic they're talking about. Before a cell is ready to divide by mitosis, it must first replicate its DNA. So within this parent cell, that 2N becomes 4N internally. Okay? And that happens in the S phase of the cell cycle, a very important step of the cell cycle. And that always must occur successfully before mitosis or this type of cell division occurs. Now that we're to a 4N state with 2 identical copies of the diploid genome, that equals 4, the cell can now divide through the coordinated action of a lot of different proteins, and mitosis can complete. So that 4N becomes 2N and 2N in 2 different cells, and now we have created the 2 daughter cells which are genetically identical to the parent cell. While we're talking about mitosis, let's zoom into the actual chromosome level, and let's do so with a specific example in mind. In this case, we're going to look at the pigeon. For the sake of this example, we'll say that the genome, the entire genome of this pigeon, is organized into 3 unique chromosomes. That is, the N number is 3 for this pigeon. Humans have, in one complete set of our genome, 23 unique chromosomes. Okay? By unique chromosomes, we really mean non-homologous chromosomes, in that they each contain their own set of genes per each of the chromosomes, and because of that, they're a different length and a different shape. Now, most of this pigeon's cells are diploid. Okay, diploid cells make up the majority of an animal or a plant's body mass. Okay, the only cells that are haploid are going to be very, very unique, reproductive, specialized cells that occur in only one small section of the body. Hold off on thinking about those for now, and we'll get to those in a moment. Let's start with a diploid pigeon cell, which is 2N. If the cell is really 2N, that means it has 2 complete copies of the genome. And we know from our description here that a single genome has 3 unique chromosomes. So if a pigeon cell is 2N, then it will be 2 times the base number of separate chromosomes, which is 3, which will give us a cell that has 6 total chromosomes. Now, this diploid cell ultimately came from a zygote after it was created through fertilization and through reproduction. And because of that, it has 2 different sets of genetic information. One I'm showing here in blue, and one I'm showing here in red. The blue set has chromosomes 1, 2, and 3 from one parent. Let's just say in this case, the father. And the red set here has chromosomes 1, 2, and 3 that have very, very similar form as the blue set, but those came from mother. The relationship between blue chromosome 1 and red chromosome 1 is that of homology. These are homologous chromosomes. This is a homologous pair. There's a homologous pair of chromosome 1s, a homologous pair of chromosome 2, and a homologous pair of chromosomes 3. Conversely, the relationship between the blue chromosome 1 and the blue chromosome 2 is a non-homologous relationship. These two blue chromosomes are colored blue because they come from the same parent, not because they have identical information on them. You can see that chromosome 1 and 2 are completely different shapes. This indicates that they contain completely different loci or genetic locations. And so while chromosome 1 in blue might have a gene for eye color, chromosome 2 in blue will not have that gene for eye color. Instead, it might have a gene for hair color. Now that we've established which chromosomes look like in a diploid cell, let's think about the diagram that I drew before, which shows DNA replication and then subsequently mitosis and cell division once that DNA has been replicated. As we go from 2N to 4N through DNA replication, the chromosome sets that you see down here are duplicated physically and remain associated with one another. This pigeon cell, after S phase of the cell cycle, is now in a 4N state. In a 4N state, this cell can proceed to mitosis because it has doubled its amount of DNA and it is now prepared for cell division down here in mitosis. You can see now that we've got a new relationship emerging, and that is sister chromatids. Sister chromatids look like these little butterfly structures, and chromosome 1 in blue here on the left and chromosome 1 in blue here on the right are identical copies of each other through that process of DNA replication, barring, of course, any unforeseen mutations that occurred in that process. Try not to get confused with the word chromatid here instead of chromosome. The reason that we call these chromatids and not chromosomes is that they're actually joined together physically at the centromere. So technically they're sister chromatids, but they are all one single molecule because of the centromere joining there. Now for this 4N set of genomes in this replicated cell, to return to a identical daughter cell state at 2N, we're going to need each of these sister chromatid pairs to separate from one another. So while the blue chromosome 1 here must split left and right into two different regions, so must every sister chromatid pair in this drawing. Okay, so the red chromosome 1 must also split, the blue chromosome 2 must split, okay, and we must get an equal segregation of these sister chromatids to end up with a 2N cell here and another 2N cell here that represent the parent cell to begin with. In the first stages of mitosis, the chromosomes will condense the way that I've already been drawing them here as these little hot dog structures. And during metaphase, they will align along a metaphase plate. The key to understanding mitosis at the genetic level is understanding and recognizing the pattern at which these align along that invisible line. Okay, so the metaphase plate goes right down in the middle here, and you can see that for all 4 complete sets of the pigeon genome in this cell that's about to undergo mitosis and is actively undergoing mitosis, that with this type of alignment, this will result in equal segregation of all genetic material, such that both sides of this divide will end up with the full complement of genetic material of the parent cell with no loss of information. We now end up with 2 diploid cells. Mitosis is what most cells in this pigeon are doing. So all the muscle cells and digestive cells and brain cells and bone creating cells are dividing this way, and thus the genetic makeup of most of that pigeon's cells remains identical throughout the life of that pigeon. The other type of cell division occurring in this pigeon is meiosis. Meiosis occurs specifically in the sex organs of the pigeon, so the ovaries or the testes is where this is occurring. And just like with mitosis, like we saw before, these sex cell precursors in those tissues start as diploid cells. They must first undergo DNA replication to create a 4N cell, which has 4 complete copies of the genome. And then at first, that 4N cell divides into 2 cells through meiosis 1 to create 2 2N cells. And then subsequently, meiosis 2 happens, and there's another round of cell division, which produces 4 haploid 1N cells. These haploid cells are referred to as gametes, as stated before, and they are non-identical. So meiosis doesn't produce identical daughter cells the way that we saw in mitosis. Let's now look at metaphase 1 of meiosis 1. So as we go from 4N to 2N during meiosis 1, let's contrast the way that these chromosomes align in meiosis, contrast that to the way that they align during mitosis here on the left. Instead of every pair of sister chromatids straddling the line and appearing on top of one another the way we saw in mitosis, in meiosis 1, sister chromatids sit across from their homologous partner. Okay, so in this case, chromosome 1s in blue are across the metaphase plate of the red chromosome 1s. And the 2s are across, the blue 2s are across from the red 2s, and the blue 3s are across from the red 3s. Right away, you can see this is going to result in a much different segregation of genetic material, and that it won't be equal segregation left and right. In this instance, sister chromatids stay together, and so perhaps the blue chromosome 1s travel to the left, and the red chromosome 1s travel to the right. The chromosome 2 sets will segregate independently. We call this independent assortment of the 1s above them. Okay, so in this case, maybe the red chromosome 2s go left and the blue chromosome 2s go right. And then down at the bottom, the 3s that are blue go left and the 3s that are red go right. Okay, any number of combinations of segregation can happen here. But in the end, you see that there's unequal segregation where the genetic material from the blue chromosomes 1 and 1 here is very different than the genetic material from the reds 1 and 1 because they came from separate parents. If we get chromosome segregation with the blue sister chromatid 1s heading this way, the red sister chromatid 2s heading this way, and the blue sister chromatid 3s heading this way, then we end up with a 2N cell, a new one, that is not genetically identical to the sex cell precursor that divided to create it. It's at this point, during metaphase 2 of meiosis 2, that our chromosomal alignment here looks very, very similar to how chromosomes align during mitosis. In this second cell division, okay, in meiosis 2, the sister chromatids will finally segregate from one another. And in this case, this is going to result in totally mixed up genetic material from the parent cell. And you're going to end up with 4 non-identical haploid 1N cells. In addition to the genetic novelty created by the unequal segregation of chromosomes in meiosis, one thing that I haven't mentioned at this point in the semester is that tracking back to over here in meiosis 1, when you've got homologous sets of chromosomes lining up beside each other, like butterflies holding hands, there's another process that creates genetic novelty called crossing over. And crossing over occurs at this stage, and it can lead to mosaic chromosomes. These mosaics are going to be chromosomes that have both blue and red sections on them that will then, again, segregate independently in meiosis II, creating even more unique combinations of gametes. I hope this 20-minute lesson has given you a better understanding of chromosomes and the DNA inside them, the different ways they can organize, and then in particular, the differences between how chromosomes look and their migration during mitosis versus meiosis. Of particular importance, I think, is the difference shown here between chromosomal alignment in mitosis, specifically metaphase along the metaphase plate, and how those chromosomes align in Metaphase One of Meiosis I, shown over here on the right. I think if you can understand how this difference in alignment reflects ultimately the difference in the cells created through these different types of division, that gives you a really nice head start in genetics as we move into the more molecular details of the course.